Cortisol is released as part of the parasympathetic response when a person experiences stress. The activation of this fight or flight response tells the adrenal gland to release adrenaline, norepinephrine, and cortisol. The release of adrenaline and norepinephrine results in an increased heart rate, increased blood pressure, and faster breathing. Cortisol breaks down carbohydrates, proteins, and lipids to provide extra energy. Besides, during a parasympathetic response, your body naturally releases extra cortisol diurnally as part of the circadian rhythm to assist with metabolism. In a fight or flight response, the hypothalamus in the brain releases corticotropin-releasing hormone to the pituitary gland. In response, the pituitary gland releases adrenocorticotropic hormone into the bloodstream. The bloodstream carries the ACTH all the way to the adrenal cortex in the adrenal gland. When the adrenal cortex receives the ACTH, the zona fasciculata releases cortisol into the bloodstream. Cortisol can be measured in 4 different ways: through blood, urine, saliva, and sweat. Blood yields the most reliable measurement of cortisol and is measured through an enzyme-linked immunoabsorbent assay, or ELISA. In a sandwich ELISA test, the chamber is prepared by coating the walls with a capture antibody. The blood sample is then added, and the cortisol binds to the capture antibody. A detecting antibody is added, which binds to the already bound cortisol. A final enzyme-bound antibody is added, which binds to the detecting antibody. Then, the sample is flushed, and tetramethylbenzidine is added, which is converted by the enzyme into a colorful output. The darker the color, the greater the amount of cortisol. Blood, urine, and saliva can be measured through another immunoassay called a lateral flow immunoassay. In this test, the sample is absorbed by the sample pad and moves linearly across the pad through capillary action. It first goes through a conjugate pad, which contains conjugates in a salt-sugar matrix. These conjugates include antibodies that bind to the cortisol and are bound with a color-producing substance. The conjugate-bound cortisol then binds to the test line, which is a row of antibodies that bind to the cortisol-bound antibodies. Other conjugates bind to a further control line, which just indicates that the test is working. This creates the familiar 2-line test used in pregnancy tests. The levels of cortisol can be measured through the strength of the color. Cortisol levels in sweat can be measured with an organic electrochemical transistor, or OECT. In an OECT test, the amount of cortisol is measured through the conductivity of the channel. An electric current flows from the drain to the source, and a charge is applied to the gate in order to attract positively charged cations. These positive cations pull negatively charged ions naturally occurring in the sweat into the channel. However, these ions must first pass through a molecularly imprinted polymer, or MIP. The MIP is specialized for cortisol, and cortisol present in the sweat will block the pathways, preventing the passage of negative ions. In other words, the more cortisol present in the sweat, the fewer ions are able to pass through. When the negative ions are able to pass through, they stunt the conductivity of the channel, and therefore the difference in voltage between the drain and the source can be used to determine the amount of cortisol present in the sample.